My name is Hiroyuki Kamiguchi, a senior team leader of RIKEN Brain Science Institute. My research field is neuronal cell biology and developmental neuroscience.
My name is Yoshio Hirabayashi. I'm also team leader of the Brain Science Institute. I'm really biochemist, especially lipid biochemistry and glycobiologist.
My name is Adam Guy. I'm a researcher in the Kamiguchi lab at the RIKEN Brain Science Institute. My specialty is developmental neurobiology.
So we started this collaboration almost 10 years ago because Dr. Hirabayashi discovered very interesting new lipid molecule. When our brain and spinal cord are constructed during development, neurons send out long axons that must find their way through complicated neural tissue and make synaptic connections with appropriate targets. In this science paper, we have discovered a new lipid that tells axons where they should go.
Initially, I didn't expect our lipids has such biological functions, just coincidence. So we have to examine many other things. One of them, this lipid has very strong guidance regulation activities.
The most important point, most surprising point of our study is that we discovered a lipid, but not a protein, that plays such an important role in our body.
So the human body, also as all other complex animals, possess many senses. Among them are nociception, which is sensation of pain, and proprioception, which is the perception of one's posture, one's body. Nociceptive and proprioceptive neurons, they have their axons coming from either the skin or the muscles, but their central projections, the one heading towards the spine, they actually enter the spine through the same place, but make connections in different parts of the spinal cord. And that's because for nociception, you send information about pain to one part of your brain. For proprioception, which is more of an unconscious process, that information is sent to a different part of your brain. Cool. This is a cartoon, it's a cross-section of the chicken spinal cord, okay, in the 6th stage of development. Spinal cord and attached to the dorsal root ganglion here, in which is contained the cell bodies of sensory neurons. In green represents nociceptive neurons. Now their cell bodies lie within the dorsal root ganglion. They extend an axon into the spinal cord via a structure called the dorsal root. Conversely, proprioceptive neurons, represented in blue, their cell bodies lie also within the dorsal root ganglion structure. They extend axons into the spinal cord via the dorsal root, so the same structure as the nociceptive axons. They enter the spinal cord together, mixed together. So you go from a mixture of nociceptive and proprioceptive axons in the dorsal root ganglion, the dorsal root, but on entering the spinal cord, they actually separate or become segregated depending on their type, whether or not they are nociceptive or proprioceptive. And our study found that the mechanism or the way that they segregate into the two different types is because of a lipid called PG, okay, which we represent it just by this red patch here. And nociceptive axons are repelled by PG, and so they cannot enter the area where you have PG. Proprioceptive axons do not recognize this repellent signal from PG, and so they project deeper into the spinal cord, into the PG domain, the PG area, and they're capable of forming their longitudinal structures. So this lipid, in essence, forms a barrier that is specific to nociceptive axons to keep them out of the area where you have the proprioceptive axons. We culture single nociceptive DRG neurons in vitro in a culture dish. Okay, we choose one axon that is growing in a straight line, straight ahead, we can actually introduce a microscopic gradient of our lipid species from an angle here. And what happens, we take a photo every 5 minutes, okay, we call it a time lapse, and we observe how the axon, as it approaches this gradient of a lipid, how it acts. It can do one of three things. It can turn towards the higher gradient, the higher concentration of your lipid, or it can turn away from it. Alternatively, the axon can simply ignore the gradient and move on in a straight line. This is the axon here. The structure at the end of the axon is called a growth cone. Okay, and you'll see that react to the concentration gradient as time passes. So within 10 minutes already, you can see the very strong response. The axon with the growth cone at its tip immediately turns away from the source of our lipid up here. So we would call this chemorepulsion induced by our lipid of interest.
In this study, we have discovered a new lipid-based signaling system that controls the development of neuronal circuits in the central nervous system. And I'm interested in continuing this research, and we will next investigate whether this lipid-based signaling system can be a therapeutic target for spinal cord injury. And I think you must be interested in the role of these lipids in different areas.
So this lipid is quite new to us. So we have many questions as a biochemist. First is how these lipids biosynthesized in the specific cells, tissues. Also, how bioactive lipids, lysolipids, is generated. So this is just an example. We have many, many questions. So we need another 10 years, I guess. Yeah.
I hope our success here will facilitate interdisciplinary collaboration to tackle intractable problems in biomedical research, such as new lipid functions in our body.